Hello, my name is Isaac Abraham. I'm a current junior in the Lyman Briggs program.
Hi, my name is Evan Nagy, and I'm also a junior in the College of Natural Sciences. Previous studies examining the westernization of the Ugandan diet associated with low polyunsaturated fatty acid, or PUFA intake, has been shown to increase the prevalence of depression, anxiety, and other mental health disorders in adolescents. With this, a decrease in the essential PUFAs, N6 linoleic and N3 alpha linoleic, downstream of DHA, DPA, and EPA, can result in essential fatty acid deficiency, or EFAD. Within our cohort, we are looking at a perinatal HIV exposure and infection that has been correlated with a lower mental health status and a higher intestinal dysbiosis. As of now, little research has examined the relationship between serum fatty acid levels and perinatal HIV exposed infected on adolescent social stress.
Our goal was to analyze the association between serum fatty acid levels with social impacts in Ugandan adolescents sorted by HIV status. Our study cohort is separated into three distinct groups: PHIV, perinatally infected HIV youth; HEU, adolescents HIV exposed but uninfected; and HUU, adolescents unexposed and unaffected. The HUU group is our control group for the study. Each adolescent in these cohorts had a blood sample taken from them at baseline. These samples were then analyzed using gas chromatography to quantify serum fatty acid levels that were then interpreted using target links. Each participant also completed a survey accounting for mental health factors under an adapted behavioral assessment system for children.
We use two different statistical approaches to break down our data. First, we use the means and standard deviations of our data to differentiate by HIV status, specifically ANOVA for continuous variables and the chi-square tests for categorical variables. An adjusted mixed effects linear regression model was used to study the association of fatty acids and social stress by perinatal HIV status using R. Here we controlled for both random and fixed effects, such as caregiver education and sex.
Resulting from our analysis, Table 1 indicates the age and number of HIV-positive caregivers are significant. Figure 1 details beta estimates on the x-axis with our explanatory variables on the y-axis. As a general rule, values present to the right of 00 indicate a direct relationship between the social experience in question and fatty acids, while values present to the left indicate an inverse relationship. Values were deemed significant if they did not encompass a beta value of 0.
Looking at anxiety in figure 1A and 1B, we see that our control group HUU's relationship between anxiety and serum PUFA levels were direct and significant at p-values less than 0.05. Similarly, there was also a significant inverse relationship between our EFAD biomarkers, MEAD, and T/T ratio to anxiety in our control group as well.
Looking next at depression, we see that our control group also exhibits a direct statistically significant relationship between PUFA levels and depression. Conversely, the PHIV group exhibits an inverse significant relationship. The same inverse relationship is present and significant for PHIV when associated alongside EFAD biomarkers.
Finally, only the control group poses a significant in its association between social stress and serum PUFA levels. None of the cohorts displayed significant results in associating social stress to EFAD biomarkers.
From the mixed effects linear regression run in R, a summary of our significant findings at p-values less than 0.05 are as follows. PHIV displays an inverse relationship between depression and serum PUFA levels in addition to EFAD biomarkers. HEU displays an inverse relationship between anxiety and PUFA levels. HUU displays a direct relationship between depression and PUFA levels. Our hypothesis was that lower PUFA levels are associated with higher adverse social experience, and social stress presents a strong association among HIV-affected adolescents. Our preliminary findings were hypothesis inconsistent with current literature, instead displaying the opposite relationship.
The difference between the control population and our PHIV and HEU cohorts may be explained by N6 levels above sufficiency, creating a detrimental effect in the control population. Limitations of our analysis are that fatty acids were measured only at baseline, and the impact of participants' socioeconomic status may induce stress.
Future research should re-examine this relationship with serum fatty acid levels expressed as a categorical variable and should investigate the interplay between diet and social experience. We would like to acknowledge the study personnel at Central Public Health Laboratories in Kampala, Uganda, for their role in collecting serum samples and demographic information, and the National Institute of Neurological Disorders and Stroke of the National Institutes of Health for funding this research. Thank you.
Thank you.